Hi everyone! This video is about telomerase, mainly on telomerase function and how telomerase extend them. So this is a chromosome. Apology for a little bit ugly drawing, but telomerase are the repeated DNA sequence located at the end of the linear chromosome. You probably learned from this class if you have an open end, then exonucleus can come in and just eaten up this open end. Therefore, the open end could be a problem. And one function of the telomerase is that it can just loop around and base pair with itself to protect the end of linear chromosome. A more well-known function, or the primary function of telomerase, is that during DNA replication, it can act as a protective cap to preventing the loss of genetic information. Telomerase act as a buffer by preventing a non-coding region that can sacrifice during replication without affecting the essential genetic material. So to better understand its function, let's look at this imaginary scenario. So there are two strands here. They are both the same length at this end. And during replication process, these two strands will separate from each other. You probably learned that there will be a RNA primers that start from 5' to 3', and then there will be a DNA polymerase just extend the 3' end, so it will be full length as the bottom strand. And on the top strand, DNA synthesis will start from left, which we will not focus on, so ... You just need to know it will extend and it will be the same length as this complementary strand. So now let's focus at this bottom strand. Let's look at this part on the right. The RNA primers is going to be removed, and now we have a 5' end here. There's no way DNA polymers can just go from this 5' end and extend to the right. And there's no other thing on the right, at least like no 3' end on the right, that DNA polymers can extend and fill in this gap. So at this bottom part, we can see the newly synthesized strand is going to be shorter than the parent strand. So let's focus right here. If this strand continue to synthesize, it will separate, and the top part will just synthesize and it will just be the same length. But now let's focus on this part. If it continues to synthesize, the bottom strand will face the same dilemma, which is they will synthesize another strand that's going to be shorter than the previous strand. So apparently, for cell division, the strand just becomes shorter and shorter. What if we have some important gene information at the part that gets shortened, the part that gets deleted? Well, you are in big trouble. However, if your end was a long repeated non-coding strand, it doesn't matter if it gets deleted or not. And that part that can get shortened and not affect your coding gene is known as telomere. It prevents the gradual shortening of coding region and make sure you doesn't lose any important gene information and maintain chromosome stability. So telomerase protect gene from getting deleted as cell continue to divide. However, with each round of cell division, telomerase gradually become shorter and shorter. This shortening process is a natural part of cellular aging, and when some cell have very short telomeres, the cell can undergo apoptosis, which is basically self-suicide. The cell destroys itself. Interestingly, some cells have the ability to reverse telomeres shortening by expressing telomerase, which is an enzyme that can extend telomeres of chromosome. The telomerase is a reverse transcriptase, meaning that it used RNA template to synthesize DNA. You can imagine the purple stuff is the telomerase. It kind of looks like a DNA polymerase or RNA polymerase. In the middle, it will hold a RNA template, and it will also hold another sequence, which is a DNA sequence of telomeres. It will basically replicate the sequence of DNA at telomere, like the 3' end, and extend it. So to better understand how it works, let's look at the following diagram. This is the original length of the telomerase, and we can see the 3' end is longer than the 5' end as we talked about before. This is a 3' overhand, and what telomerase will do is that telomerase will come over, it will hold RNA template, a piece of RNA sequence as shown here in blue, and it will base pair with the 3' overhand. This 3' overhand are extended using telomerase RNA as a template. Telomerase shift forward and binding to newly added repeat. And this step will just continue, it will keep extending, and then the telomerase will shift over again and keep extending until you have this big long 3' overhand. After you have this long 3' overhand, at the bottom part, DNA polymerase will extend an RNA primer to synthesize a complementary strand. But remember, you will always have this 3' overhand. So basically, as you see, one strand was lengthened by the RNA template with telomerase, while the other strand was filled in by normal DNA polymerase mechanism. So as we talked about before, telomerase shortening causes aging. So telomerase is like our cellular fountain of youth. It helps maintain the length of telomerase. So couldn't we just always activate this cool enzyme and our cell will just live forever? Unfortunately, yeah, that's not the case. So just think of what is a cell that can live forever. Right, cancer cell. So that's important to realize that telomerase is not activated in most cells of our body. Its activity is tightly regulated to prevent uncontrolled cell growth, which can lead to disease like cancer. In summary, telomerase play a crucial role in preserving our gene information, and telomerase help in extending the length of telomere. That's all about telomere. Wish you enjoyed this video.